Hello, everyone. Thank you so much for being here. My name is Janet Doan. I'm the Admissions Outreach Coordinator here at Keck Graduate Institute. Thank you so much for joining our Biotech-based Therapeutics webinar. Today, we have a special faculty member with us today, Dr. Larry Grill. He is the Dean of Research and the Research Professor here at Keck Graduate Institute and will be speaking more about the biotech-based therapeutics. Again, if you could please mute yourself, that would be wonderful. And I look forward to what you have to say, Dr. Grill. I'm going to hand it over to you.
Okay, great. I see some of the students I just talked with earlier today, and welcome back. I'm going to go a little bit more into cancer therapies and primarily new biotech methods. And the reason why this is so exciting to me is because I've been around and watched everyone say, hey, I think we're getting to know cancer, we're going to be able to cure it. And we've made incremental jumps. And they've been really fairly small, mostly only allowing an additional 2 months in your life, but not really curing cancer. We now have finally, for a change, start having the ideas on how to really cure cancer. And
true, but it all.
Oops. And one thing that's exciting is there was a publication earlier this month of a clinical trial in which all 12 people in the clinical trial were 100% cured of a late stage cancer. And this is because we actually kind of know what to do on that. So I'm going to go over some of the new biotech therapies to treat cancers that are working and how come they're working and why we are really good, are on the precipice of actually doing some real cures to lots of different cancers. Instead of incremental, slightly better life or slightly longer life, we're actually finding ways to actually stop them. So I'm going to go through a lot of those. So let me first, and during this whole presentation, interrupt me at any time, but I will try to answer them as needed. So let me first share screen. Okay. And there we go. Okay. We go to. All right, so there's lots of new biotech drugs out to treat all kinds of diseases. We have gene therapy going on. We have a lot of other things going on, but I do want to just really focus on cancer, the new biotech therapies for cancer, because this is kind of a real breakthrough in the last 3 years. I know many of you have heard the word CAR T-cell therapy. It's actually pretty phenomenal technology. And checkpoint inhibitors, maybe some of you heard of checkpoint inhibitors, and I'll talk about those. But let me first introduce a little bit of numbers on cancers. So just cancer strikes 1 in 3 people in the US. It kills 1 in 4. And that's because, and I mentioned this to my students earlier today, that's because every single cell in your body, except every nucleated cell, has the potential for cancer. And why do they have the potential for cancer? It's because that's where they came from. So before we were multicellular organism, we started out as a single cellular organism. And the only thing that we had to control was finding food. And if we found food, we would grow. And that's what all your cells, every single cell in your body with a nucleus, wants to keep growing. But as we became multicellular, we had to install rules because we could not have all the cells to continue growing without controls. So we started putting in a police force and enforcing rules, turning off genes and other things like that, so that our cells would not go to their, as we call this, atavism, going back to a very early primal state where they just want to replicate. And so we actually, in cancer, a lot of the cancers are done because we do not have all of we have some of our genes turned off that are supposed to be enforcing those rules, stay as a multicellular organism. So the medical term is a malignant neoplasm, neo being new growth. And basically the major things we're going to see in cancer, and I'm going to really talk about not benign cancers, but metastatic cancer. So we're going to have uncontrolled growth where they now, instead of being regulated as a cell growth, they're now saying, all I want to do is grow. Just if I have food, if I have blood, if I had oxygen, I'm going to grow. And that's uncontrolled growth. And that's when we would call it cancer. If it's allowed to leave that current area, and that's we call that metastatic, and it moves via the lymph or the blood to other parts of the body, that's when they are now in a new zone, and now they're totally out of control. They don't even have their normal surroundings to inhibit them. So we have lots of things going on in this. Some interesting numbers. So your body, depending on your size, has approximately 100 trillion cells. That's your cells. By the way, you have about another 100 trillion cells that are your biome. So on your skin, in your intestines, all those, you have another set of 100 trillion cells. But of your cells, your eukaryotic cells, you have 100 trillion cells. About 100 billion of them go through cell division every day. That means 15 million new cells per second. Now, if you think of the population, I guess in November, we're supposed to have the population of the Earth be somewhere around 8 billion. If you think, 100 billion cells go through cell division a day, you're going to have some screw ups and something's going to go wrong. So basically, it's your cells that have to control what they do so that they don't actually develop into cancer. Because basically, you have precancerous cells every day, possibly even 1 million per day. But we have plenty of ways to stop that. So one is where we have, as I mentioned, you have to have these multicellular organisms. You have to have kind of an enforcement of rules. And these enforcement rules are things that we call tumor suppressor proteins. And these are basically the police force that go around inside your cells and make sure everything went according to the rules. And if it's not, they basically say, go to the kind of the head sergeant, we call him P53, and say, hey, something screwed up here, pull the chain, we're going to die. So this cell is going to go away. And that happens millions of times per day in your body. And that's the proper way for them to get rid of cells that didn't work. We do have a backup system. So we have our immune system. We have the natural killer cells, which keep going and checking every one of your cells all the time. And if they see some that looks like they're replicating irregularly or having some new proteins being made that they shouldn't be making, they will kill those. So basically, we're doing a really good job at controlling cancers. But we do all have some mutations that go on that accumulate and over a period of time, and it may be 100 different mutations. We don't know exactly the number you need to where the cell finally is out of control and the police force is not able to do it. And a lot of times our police force, our tumor suppressor genes are basically shut down or turned off because of our diet, because of the environment, because of several other reasons. And every time we lose one of that police force, it weakens our ability to control our cells because they keep wanting going back. Again, atavism, they want to go back to their old native state. They have all the genes to replicate totally 100% of the time. And there's something that many of you have learned, that's called the Hayflick Limit, which means your cells are only allowed to replicate somewhere between 40 and 70 times. And that's because they get some of their telomere of their chromosomes removed each time they replicate. Every time this happens, then they can't replicate again. But turns out in your old atavism cell, basically that still has a telomerase, which will keep adding it back. So we still have those things hidden in there to allow the cancer or the cells to grow totally through it. Some of the genes that are involved in cancer, there are some genes that actually in their own can actually stimulate cancer if they're unregulated. So a single gene that's unregulated, we do know there's some genetic links. There are some genetic diseases, genetic cancers, in which it's a single gene that will end up causing that cancer. But that's pretty rare. But we do have oncogenes, onco being cancer genes, so causing cancer. And most often those oncogenes are a gene that should be turned off but is suddenly turned on. Then the other genes that we have that are really important and even more important in some ways is the tumor suppressor genes. And I will go back to this organized multicellular organism. We have to have our police force that goes around and keeps every cell doing what it's supposed to do cooperativity, and so enforcement of cells that become errant and do something aberrant and do something they shouldn't do. The tumor suppressor genes say, okay, either you straighten up or else I'm going to go to P53, and the P53 is going to make our cell die. And so that's a really, really good thing. We also have caretaker genes, just take care of everything, DNA repair when something goes wrong. And these usually do a pretty good job, but it turns out really when a cell goes to be towards a cancer state, it wants more mutation. So it likes DNA repair to slow down or be more ineffective, because the more mutations it can get, the more chances it can evolve into a full-blown cancer. So those are just some genes. History of cancer. So we've known about cancer forever, but in the very early days, of course, many people died before they ever got cancer. But we have had some cancers where you've had a big growth, and obviously breast cancer is going to be one of those. And we had a primitive mastectomy in which a slave actually cut off the breast, and that because it was so cancerous. Needless to say, that was pretty dangerous before we had antibiotics. It's pretty hard to stop an infection in that way. So we've had to actually do other things. And finally, we've now in the somewhere around 1935, we started coming out with our first antibiotics. But prior to that, even if you had cancer cut out, you were more likely to die because of some kind of early infection. Then with Madame Curie and others, we realized radiation could specifically kill cells. And so they've been using radiation to target and kill cells. Now, radiation cannot, if it's in low dose, can cause cancer. In a high dose, it'll kill the cell. And so if you can control where you put it, you can use radiation. And that was done in the early 1900s. We still use very specific targeted radiation therapy. After some of these, now look at 1986. So 1986 is going to be kind of a big banner day. We're now starting to do surgery with good antibiotics. We're using radiation very pointed. We're using chemotherapy that we can really kind of kill all dividing cells. Now, killing all dividing cells is a nasty thing. You know, you lose your hair, you use your fingernails, all your biome, in your gut, all those are killed. So having all replicating cells is not necessarily a good way to do it, but it's still probably one of the most heavily used one now. Herceptin was the first biotech drug, if you will, and that's where you would use it. And I'll describe a little more how we use this, but it is an antibody that binds specifically to a receptor on the cancer cell to let your immune system know this is a cancer, we have to go after that. So that's the beginning of using immunotherapy. And I'm going to be talking a lot about immunotherapy because this is super. This is our first understanding. We hardly even understood the immune system 50 years ago. Now, not only do we understand it, but we're understanding signals that they're sending back. We're finding out how cancer cells are saying, oh, I see that signal, but I'm going to find a way around it. So we've gotten cancers being smarter, and we're finding out how to teach our white blood cells to fight better battles. And I'll go over some of those. We've also been able to start using genetics. And while we've been identifying a lot of genes associated with cancers, targeting those genes has not been successful at curing the cancer. So we really have to find some other way. You can go in and maybe do some genetic manipulation someday, but right now, just knowing the cancer, knowing what's causing it, is good for us to classify it, because even though we classify cancers by where they originate, cancers really have their own way of doing it. So they can originate in your breast, they can originate in your skin, they can originate in your lungs. There's a lot of different places, but in truth, a lot of these share the same exact traits. So once we start learning what are these traits, what is it that we can target? How do we get our immune cells to wake up and go after something that they're supposed to know how to do? Okay, some new strategies. So using molecular genetics, looking at the genes, we're starting to look at that pretty heavily. Now in City of Hope, they're basically doing a genetic diagnosis of everyone who has cancers to see if they can start putting things in, looking at the cure rates using different technologies, and seeing if they can start getting better strategies to go after cancer. It's not saying knowing the genetics is going to help you target it, but it may help you to put it into a class in which you have found some way to cure it and do that. There's another term here, blocking angiogenesis. If you were a small class, I'd ask someone to explain what angiogenesis, but I will go over that on my own. Angiogenesis is basically stimulating blood supply, so new blood vessels. Anytime you have a cancerous growth, it needs food and lots of it, and it needs oxygen and lots of it. And that's because our cells have gone to a different set of burning oxygen. It's less efficient, burning sugars. It's less effective. It grows faster, but it's using tons of energy. So it needs lots of sugars, lots of blood supply. So if we can stop that, and I'll show you the drug, one of the first ones I'll be showing you, is if we can stop those blood vessels from supplying it to the cancer cells, that's a strategy. I'll talk about oncolytic viral therapy, and we actually trained some viruses to specifically go and kill only our cancer cells. So viruses are very capable of killing cells. Many of you know that they killed a lot of the SARS-CoV-2 virus killed a lot of your cells when they targeted to when you got the COVID-19, for those of you who got it. But what we want to do, and what the oncolytic viral therapy does, is it uses a virus to target specifically only cancer cells. So instead of binding the ACE receptor, which is on most of our cells, it binds a receptor that's only found on the cancer cells. So it's really a neat strategy. Also, again, the immunotherapy, stimulating our native immune system. Our system is pretty damn good at getting rid of cancers, but sometimes the cancers worked out a strategy and found a way to communicate to your cells. So our cells have what's called a two-factor authentication. So when a white blood cell sees what it thinks is a cancer cell, it says, okay, I'm going to kill you, but then it needs to get another level of authentication to kill it. So it always has to have that. So it has communication between a couple different cells to get that. If the cancer cell learns about that two-step authentication, it knows how to get around it. And I'll talk about drugs now that we know how to block that back signal from the cancer. The other thing is these monoclonal antibodies, and the list of monoclonal antibodies that treat cancers is in the hundreds now. Really amazing. So it's a #1 strategy for going after cancers, but we're actually getting smarter ways of using antibodies, and I'll go into those as we go into it. So we're going to have what's the one using the antibody, where we call it, this is a chimeric antigen receptor, the CAR T cell therapy, where we actually genetically engineer our cells to go specifically after the cancer. And then we have a chimeric antigen receptor for macrophages, because macrophages are basically your first line of defense against intruders and some cancers. And so what we're doing is teaching them using genetic engineering to go after very specific cancers. Then the other thing we're trying to do is promote apoptosis. So apoptosis is a normal thing going on in your body. It's when cells have become anywhere near out of control, your T cells or your natural killer cells basically go and say, hey, I think you're out of control, will you please kill yourself? And so the cell says, okay, and it in conjunction with the P53, it goes through apoptosis. And so what we will need to do is make sure that these cancer cells recognize it and go through apoptosis. So in most of the cancers that we see, that P53 gene has been inactivated. So cancers do not like P53, because P53 says things are not going right here. We need to go through, we need to kill this cell. And basically, if they've disabled the P53, then that cancer cell is a happy cell, because there's no protein that's going to say it's time to kill ourselves. The other thing we're going to, there's a new drug that's out there that says, let's just stop metastatic. So if you can keep it in a certain area and you don't let it go out, that's great. A benign tumor can be removed. If it doesn't go anywhere else, no big deal. And most of us don't worry too much about a squamous cell carcinoma. We want it removed because it's a tumor growing right there on your arm, doesn't look good, but it's not metastatic. So you don't worry so much about the metastatic. So really, that's another route we're going to go after it. So there's a lot of people who are sequencing cancer genomes, and I mentioned City of Hope, and looking for different classes and looking for how the current drug technologies are doing them. And I'll come to one that's we found that there's a lot of cancer cells that have HER2/neu as a receptor on the surface. We've known in early days that breast cancer, at least a certain percentage of breast cancers, has a high level of this HER2/neu receptor on the surface. And so we know how to target that. But we've now found that there are several other cancers that have a low level of the HER2/neu. And so we've had to get smarter and go after those cells that have a low level of HER2/neu. And so a new drug that just came out is capable of going after the low level HER2/neu, and that's going to cover up to 50% of breast cancers instead of the 15 to 20% that the earlier one called Herceptin was able to do. Also, combining treatments is something we can do. Once we've seen that we've been able to address this particular aspect of one, let's use that drug and another in combination. What we don't want is we don't really want our cancer cells to work its way around one drug. We want it really to work it around a whole bunch of different blocks we put up. So going into some of these, so here's angiogenesis. So basically, you're going to have your blood vessels, and anytime your cancer starts growing, they know how to actually trick your immune cells into setting out cytokines that ask for new growth of these blood vessels. That's a pretty good talent that they have. Again, back to that atavism, that they got a pretty neat way of figuring things out to get what they need. And so we now have drugs that will actually just block that so that we can't start making the new blood vessels. So these are some of the inhibitors of angiogenesis. The first one was put out by a company called Genentech called Avastin. And then we've had some others that have come up that also block this in one or different way. And that's been successful, and Avastin's been used in several different cancers. It was actually also approved for breast cancer, but after it had been used for a while, they saw that it only shrank the cancer, but did not extend the life because there was still too much metastasis going on. And so the FDA said, okay, all you're doing is making it look like they're getting rid of cancer. You're not. So Avastin is no longer used in breast cancer, at least on label. So doctors aren't allowed and insurance companies won't be paying for it because it just hasn't shown any efficacy. So you have to be careful of what you use these drugs for. Okay, I mentioned that basically we do know viruses are very good at killing cells. They've been doing it for a long time. They know how to do it. When a virus goes into the cell, all it wants to do is make more viruses. So it has the genetics to get that cell to start making more viruses. But usually they will kill the cell because they don't care. They just want more viruses. So what they've been working on is finding a way. So remember, and I go back to SARS-CoV-2 because everyone knows about that particular virus. We know that we have a spike protein that binds to the ACE2 receptor on your cells. Well, all viruses in the mammalian system have to basically be able to bind to a receptor. So what they've done is they've genetically engineered a virus so that it goes into only a certain receptor that's on a cancer cell. And we've genetically modified this herpes simplex virus, and it targets the cells that are overexpressing an HPV receptor that we know about. And it goes in, the normal cell, it goes in, it doesn't replicate, so no big deal. If it goes into the tumor cell that has the right environment, it will start replicating, kill the cell, and then it has offspring that will go to nearby cells. So it's a self-amplicating anti-cancer virus. And in theory, we could design viruses to go after lots of different cancers. So this is the first one approved, and I see that we will probably have more going because viruses are pretty good at getting into cells. They've been doing it for millennia. Very good. They can go into every cell. Bacteria, fungi, algae, everything. So we know this is, they have great capabilities. In addition to killing that, it basically activates your immune cells because this is a viral infection. Your immune system does not like virus-infected cells. The natural killer cells and the killer T cells both are running around checking on the outside of something called a MHC presenter that's letting the outside world know what's going on inside that cell. So they put samples of things that are going on out on the surface, and that's called your HLA presenters. Everyone has a great way of doing this. But anyway, so we do have these basically these receptors putting out pieces of this virus. And the immune system is now activated to go after the cells that have been infected by the virus, but it's also been putting out pieces of that. So you're getting your B cell system and your T cell system going after parts of the virus infected cell that have kind of been destroyed. So we're now activating using a different route. We're activating your immune system to go after these cells. So really great way to start going after tumor cells. And again, very successful therapy that has just come out. Okay, so stimulating our native immune system is absolutely the way to go. We've been messing around with lots of different things, but now we're starting to understand we need to tell our immune system how to kill that. And we've now found lots of different ways of working with our own immune system. So one of the ways that our immune cells find cancer is the cancer cells will start producing proteins that they're not supposed to. Okay, so they're going more to their atavism, their an early state making proteins they're not supposed to make. And these are put on the surface, and any of your natural killer cells go and kill any of these cells. And so basically, this works pretty well. But now that some of these cancers have found a way around it, we have to find a new way to target them. So it turns out we have another system in our immune cells in which if there is an antibody that's binding to a cell, it's the back part of it. So if you actually think of, and I'm going to kind of draw with my hand, if you have our antibody with the Y at the top and the bottom part is our constant region. If this top part is binding to a cancer cell, the bottom part, the constant region, is dangling out into the circulation system. And you have white blood cells, macrophages, that specifically say, oh, wow, there's that constant region binding to something. And it goes and checks it out, and most often it will kill it. So it has several different ways to kill that cell if it has that antibody binding to the antigen on the surface of that cancer. That's why we call them kind of cytotoxic T cells. They will see that bound and they're going to attack it as long as they have that specific antigen. That's why we have literally hundreds of cancer therapies. They're based on monoclonal antibodies that will bind to various specific receptors on or surface proteins on cancers. And again, that's to re-stimulate your own immune system to go after something that it's forgotten how to do. So we're just adding a little stimulus, and it's working quite well. The first one that really worked well on that was Herceptin, and it was pretty effective at killing cancers for the women who had high HER2/neu receptors on the surface of their breast cancer cell. Unfortunately, that was only between 15 and 20% of the women who had cancer. So we're still missing quite a bit of those. Okay, the other thing that they've done in therapy is they've actually taken the cancer cells, blended them up, and injected them back into your body. And those pieces have stimulated your immune system to go after anything that has those pieces of the cancer cell. And that's a little more dangerous because you could end up putting in a piece that you don't want to go after. So, but we have tried those therapies. That's not one of the ones that I'm going to really go into because it still isn't one of the successful ones that we have going there. Okay, so here we go. Stimulate the innate immune system. The earliest way, naked antibodies. Just make the antibody, inject it into the body. It goes and binds to the cancer. Now your macrophages and your killer T cells are reactivated because they now see this dangling constant region hanging onto a cell. And it's kind of like a fishing lure. It pulls them there and they say, oh, yeah, this is cancer. Let's go after it. So they can work by themselves. But we've actually become a little more sophisticated. We've actually added other things to it. So we've actually added things where we take a chemotherapeutic drug and we attach it to that antibody. So basically the antibody goes to, say, the HER2 new receptor, binds to the breast cancer, but instead of just relying on your immune system to help out, it's actually carrying something that is going to kill that cells. So we have something that you take some radioactivity into it and use radioactivity. So it's a low level dose of radioactivity, but as these all bind to that cancer, suddenly you're getting a higher level of radioactivity. So it's very localized containment of radioactivity to kill the cancer cells in that region. The other thing we have here, now this is Kadcyla, is an improvement on Herceptin. So that's going to be the one that binds the HER2 new receptor on breast cancer cells. And they've taken that in. So this is the next generation of Herceptin. And it goes in, it binds, and then it kills the cancer with a drug called DM1. So it's a very localized, we're killing not all the dividing cells, but all the dividing cells right there by the cancer in which that antibody has bound. So another sophisticated way of going after it. I mentioned that there was something that was even better than that Herceptin. And basically, we know that HER2 is on about 10 high levels of HER2 new are on the 10 to 15 to 20% of breast cancers, but there's a low level of it on many of the other cancers and up to 50% of breast cancers. They've actually found in HER2, which is also taking a drug that kills the cells, but they found they can bind to lower levels of HER2 new and basically kill more of the cancer cells than before. Not only that, but we've also found that HER2 will bind to other cancers. So while we do have it in breast cancers, we found low levels of HER2 new can be in gastric, lung, colorectal, ovarian, and prostate cancer. So now, again, knowing the genetics, knowing do we have HER2 new on that low levels of it? If we do, doesn't matter if it's in the lung, doesn't matter if it's colorectal. Let's go within HER2. And this drug has just been approved within the most very recent time. So Herceptin is the old one, and HER2 is a new high-tech one that we have come out with. We also have bispecific monoclonal antibodies. So bispecific, it means that it's able to bind two things. So this does another trick to get your immune system to do what it should do. It's going to use one arm of the Y to bind to the cancer, the other arm of the Y to bind to a specific T cell. So the T cell comes in close proximity of the cancer cell, and bringing them in close proximity, now the T cell says, oh yeah, I guess I better kill this. So the T cell does what it does. So we have several different new bispecific monoclonal antibodies, which brings our immune system to take a look at this cancer by attaching to the CD19 protein on leukemia and lymphoma cells, and then it binds to the CD3, which is on T cells. So by binding them, we get an immune attack and go after the cell. So neat trick, fun to make it. Here's one of the drugs used for that. But it's going to bispecific. Here's our T cell, here's our tumor cell, brings them into close contact, and we have really good survival rate of using this bispecific antibody. Okay, this is fun. So I'm sure all of you have seen a chimera, and you know what a chimera is. In Greek mythology, here we have the goat head, the lion's head, the snake's tail. Basically, it's really quite interesting where you have things put together to make something that's dangerous and scary. Well, we're using this in fighting cancer. And so the way we do it, we call it chimeric. So again, our chimerica antigen receptor. So this is where you get that CAR. And again, we're using T cells, so it's called CAR T cell therapy. This is really pretty neat because this is where we're really going down to the immune system, finding out what's been going wrong. Why isn't the immune system killing that cancer? What do we do to change the rules to make it, to get your, oops, let me go back, to get your T cells to go after that particular T cell therapy. So these are, again, I'll show how we make these, but we're going to basically develop these to target cancer cells. The first ones were out were called Kymriah and Yescarta. Yescarta was done by Kite Therapeutics, and it was bought up by Gilead. And it was one of the first ones. Actually, Yescarta, I believe, came out a little bit earlier, but against leukemia and a lymphoma. And basically what we're doing is we're designing this and training your white blood cells to go after the cancer. So this is the design of a chimera. So we're actually taking and genetically engineering a protein. So on the outside, we're doing basically an antibody kind of surface that's going to be able to bind to the cancer cell. Then we have a transmembrane linker, and then down below we have this is the other part that's from a different thing. We're adding in a signal that anytime this binds, it's telling the cell to release everything it has to kill whatever's to kill. So basically, when this has been activated, we're going to kill. So forget that two-factor authentication. If it binds, it's going after that cell. So now we're saying, okay, we're starting to learn. Let's go after the cancer specifically. We've had approval of 6 or 7 now, and I believe there are literally probably 100 in the pipeline for the FDA in clinical trials. But it's a super neat trick to do this. So the CAR T cells that have been targeting CD19, which is on several cancers, have shown to be very effective. Again, we're having this chimera that binds to the CD19, and then it tells the cell, hey, I just bound to something, you kill it. And so they release all their different perforating proteins and other proteins just to kill that cell, and they're pretty good. Now, yeah, are there some slight problems? Yeah, we have something that's called cytokine release syndrome and another one. But if you're on this therapy for about a couple of months while you're taking the drug, you need to be in close proximity to the hospital because they know what to do in cases of this. So we're going after cancer. It's very sophisticated, but we're using sophisticated ways to go after the cancer. So again, it's really neat. This chimera is something that we designed to specifically kill specific cancers. And in theory, we should be able to target any cancer cells if we can find the surface protein. And that way we can avoid that two-factor authentication and just go and kill anything that has that on it. Now you're saying, why do we have 2-level of authentication? And that's because we always want to make sure your T cells are under control. In other words, that they just don't go killing anything that they think is going to be a cancer cell, because it might be a good cell. And when we get something that's going a little screwy on that, we sometimes get autoimmunity. So your body really fights this level between autoimmunity and killing the cancer cell. Once we bypass us with our chimeric, that's because we actually know what we're doing, and we said, here's what you need to do. We've actually gone to another stage, and that's called a chimeric antigen receptor using macrophages instead of T cells. So it's CARMA instead of CAR T. And basically what they're using in the case of the CAR T cells, they're using a defective HIV virus that can't cause disease and can only integrate the protein for the chimera into the chromosome of the T cells. They take it outside of the body. They so basically, if you go in for CAR T or CARMA, you're going to go to the hospital or the clinic. You're going to have your blood taken out. You're going to take out your T cells or your macrophages. They're going to return the rest of it back to your body. Then they're going to genetically engineer it with the new chimera, so that you now are getting your sophisticated training, if you will, of your T cells or your macrophages. They repopulate them outside of the cell and then reintroduce them back to you. So these are really your own cells that are coming back into you, but now they have become very targeted soldiers. They're going after one thing and one thing only. And that's because you now train them using this chimera that has been set up. So these are the ways that we do it using the CAR T and the CARMA receptors. Okay, let's go back to, I talked about the two-factor authentication. So basically what you're gonna, so this is gonna be a T cell right here. Here's our T cell receptor. It's binding a tumor on the cancer cell. So I mean a tumor particle on the MHC presenters of that cell. So it recognizes this as a tumor cell, but it needs a second level authentication. And we call this programmed cell death receptor. So PD, programmed death. And that's kind of your two-factor authentication. And I kind of call it a secret handshake. So this tumor cell has learned that if it knows how to do the handshake, the second level authentication, this T cell will say, oh, sorry, I didn't know that. And that T cell will turn around and leave. And the cancer cell just jumps up and down and says, yeah, I know how to do this. So it knows the secret handshake. So some of the new therapy says, okay, now that the cancers have learned that, let's block one side or the other. And so we've been able to do that. So some will block the PD-L1 of the tumor cell, and some will block the PD of the T cells. So as long as you stop this secret handshake, then this T cell is going to go ahead and kill the tumor cell. And so these checkpoint inhibitors have been really pretty neat and pretty sophisticated. Very new technology. And there are several different drugs that do that. So anyway, so it's going to be T cells, cancer cells. So you're going to go after each one. So this is one of them. It's called Keytruda. And it binds to the PD-1, which is going to target your immune system. So it's going to your immune system. It's going to block that PD-1 so that the cancer can't do that secret handshake. So the T cell doesn't know it. It says, hey, I'm not getting the second factor authentication. I'm going to kill you. And so that works pretty well. And that's Keytruda, done really well. It's been approved for melanomas, non-small cell lung cancer, squamous cell carcinomas, non-Hodgkin's lymphoma, and we have other additional clinical trials going on for this anti-PDL. And again, you're just going after the second factor authentication that the cancer cells have learned and blocking it. And now the cancer cells are going to be killed because even though they know the secret handshake, they can't get to the hand to shake it because the antibody's binding to that. Okay, this is just the mechanism of action. I kind of described it just well. Here's our PD-1, how we're blocking it with Keytruda, the antibody. And even though it's recognized the cancer, it was waiting for a secret handshake. It didn't get it. So now it's going to kill the cancer cell. T cell's going to get it, go after that. Abdivo is another one that's out there, approved for advanced lung cancer. And again, this is blocking that receptor. Then we have something, and this is the one I just talked about very early in this talk. So FDA approved June 5th, 22. So that means this month. This drug, there was a paper published on this new drug, and it's going to be a PD1 inhibitor. It's called Jemperli. And again, it's a monoclonal antibody that's going to bind to the PD1 on the T cells. And basically, these people were in stage 2 or stage 3 rectal adenocarcinoma, likely going to be dying. And astonishing, it was a small cohort. There was only 12 patients, but all of them, the cancer went away, totally went away. And as the author said, I believe this is the first time this has happened in the history of cancer. The fact that 100% cure rate of the cancer, just by blocking that secret handshake that the tumor cell learned how to do. Here's another one, an anti-PD-Well1. This is going after the cancer, blocking the hand from the cancer that's supposed to shake the PDL on the T cell. Now that it's bound, then that cancer cell is going to be killed because its T cell has been blocked. I mean, the secret handshake's been blocked. We also have other cells. So we have dendritic cells, and they have similar checkpoint inhibitors. So we also have anti-CTLA, so it's a little different. So with the PD1, we have that going, inhibiting here. With the dendritic cells, we have anti-CTLA 4, which is basically a checkpoint inhibitor also. So we're starting to learn enough about how cancers are getting around it that this is now super way to go after it. Okay, finally, I want to just go back to that P53. So I mentioned earlier that this P53, which is one of the it's kind of like the master tumor suppressor gene. It's the one that is kind of like the master switch on a electrical box. It pulls the switch down when it sees something went wrong in the cell, says time to die. So that's controlled apoptosis, total cell death. And basically, that's in the majority of the cancers, over half of the cancers, P53 has been inactivated. And that's because cancer cells do not like P53. They do not want to go through apoptosis. They want to survive. So basically, they've inactivated it in some way or another. So basically, this P53 protein, we got to turn it back on, but we don't know how to turn it back on. So again, here 50%, 70% of the tumors, it's been either turned off by epigenetic forces or mutated or even deleted. So what has been done is they have found that if they can reestablish the P53 in cancer cells, they can actually get apoptosis turned back on, because the P53 still recognizes and says, hey, I was gone for a while, but I'm back and I'm going to kill this cell. So there have been a couple of drugs that have been approved in China. They're in clinical trials now in the US. These are the names of it. But basically, they're using gene therapy where they're introducing a new gene to the cancer cells. So the cancer cells have turned off that P53. This gene therapy brings that P53, puts it back into the cancer cells, and the cancer cells die. And that was not what they wanted, but hey, they've been genetically engineered to go back with that P53. I want to skip through this because there's been clinical trials showing a pretty good result in that. Again, we're going to see that in the United States. But I wanted to make sure I left time for questions because I know I've talked a whole lot and I went fairly fast. So I'm going to stop sharing and I'd ask for any questions that you have out there.
Please feel free to unmute yourself or type the question in the chat. Thank you so much, Dr. Grill, for the presentation.
Sure.
I had a question.
Sure. Go ahead, Katie.
Hi. I know that some people are like genetically predisposed to having breast cancer. I was wondering if the gene that they test for, does that just have a tendency to turn P53 off? What does that gene do?
So yeah, it's a good question. So we know the BRCA1, BRCA2 are associated with a higher level of cancer. There's no proof that it causes as in responsible cancer. It's an association that we know of right now. So, guilt by association is a pretty good association, but we don't know exactly how that leads to the cancer. We just know that if we look at it, you have a higher chance, not 100%, you just have a higher chance. And so a lot of people, as you know, have elected, if I have that, I have a higher chance, and I just don't want any chance. So let's get rid of the breast. I think that's an interesting way to do it. I would rather see us go after a target, or else find a way to stop it, like with this metarestin, which I didn't go into detail on, stop metastatic, because we know metastasis goes on very early in cancer stages. And it's hard for it to reestablish anywhere. I mean, you have to realize a cell that breaks off and gets thrown into the bloodstream and lymph nodes, everything has a lot of T cells out there looking for that. So it has a hard time getting around, and has a hard time in a new environment. So most often the metastatic cells don't work well, but once they do get established, it's dangerous. But if we can find a way to stop them from being able to even turn on, that's a route we can go. But again, knowing that BRCA1 and BRCA2 are a link, maybe someday we'll get to know what is it that allows cancers to come. Is there something we can block later on, as opposed to doing a preventative by removing the breast? So I look to that in the future, because we're learning so much and it's going so fast. Most of these drugs I've just talked about have been within the last 5 years. So the Pd1 inhibitors, the CAR T cell therapies, the Karma hasn't been approved yet, but all these different things are just brand new, and we're just leaps and big leaps in going forward to being able to use biotech and our brains knowing what to do to get our cells to kill the cancers.
Thank you.
Thank you. Are there any other questions?
I see in the reference to CAR T cell that said, what stages of cancer can that treatment be used? Clearly, the earlier you go to, you can, but basically, most often it's being used at phase or at the stage 3. But in theory, if you can really go after it earlier, you're better off. But at stage 4, it's still possible to even go after cancers using the CAR T cell therapy in stage 4. One of the early things is we can start detecting in the bloodstream beginnings of metastasis of other cells going where they shouldn't. That would be getting those early stages. We'd have a really good chance of getting control of these metastatic cancers very early. Other questions? Either I was, Nahir.
Yes, I wanted to ask, I believe that the name was Keytruda. The interaction was between the tumor cell and a natural killer cell?
Yeah, so basically Keytruda is going to be, let's see, Keytruda was actually bringing in, I should go back to that. Let me see.
We're talking about the secret handshake.
Oh, the secret hand. Oh, yeah. So right.
Yeah.
So Keytruda was going to be binding to the anti-PD1 on the tumor itself. So it basically is we know that the tumor has learned what the secret handshake is and puts out its hand. And what we've now done is a monoclonal antibody which binds to the hand that wants to reach out to do the secret handshake. That's what you're asking?
Yes. So basically the natural killer cell recognizes it like a self cell, so it doesn't activate. So
if what it's done is it's recognized it as a cancer cell, but the cancer cell with its two-factor authentication reaches out and says, no, I'm really not a cancer cell. And basically the T cell says, oh, okay, so it turns around and goes away. So what we've done is that second factor authentication that the T cell really needs to kill has been turned off by that monoclonal antibody binding it. So now it won't kill that T cell.
Okay, perfect. Thank you.
Sure. Other questions? Okay.
All right, there is a question in the chat, but I believe it's for you, Dr. Grill.
Yeah, so many of you know, well, some of you know, in the past, pre-COVID, I used to take students to Botswana to learn the ways of doing developing country research and how to get, in specific, working on vaccines. With the COVID outbreak, we had to cancel it for the last 2 years, and we were trying to get it to go this year. It did not. So I'm hoping that we have and we called it the Be Sure program. I'm hoping that we have it back up and running next year. It's just they are a little more bureaucratic, and it was harder to get things going. You need almost a full year to plan and to get everything organized. So I'm hoping.
Thank you so much, Dr. Grill. And thank you everyone so much for all of your questions and your participation. I really appreciate it. If you have any other questions, please feel free to email me. I'll put my email in the chat. And thank you so much for being here. Thank you so much, Dr. Grill, for the presentation and for your time. We all really appreciate it. I hope you all have a wonderful rest of your day, and I hope to see you soon at KGI. Thank you so much, everyone.
Thank you.
Thank you. Thank you, Dr. Grill. Bye.
Sure.
Again, thank you so much, Dr. Grill,
Sure.
for working
All
with
right.
me on this. All right, bye.